Good afternoon, ma'am. Good afternoon, classmates. I am the first reporter, Sherilyn Q. Pelayo, and my topic is all about oxidative pathway, hexose monophosphate shunt. First, I will introduce my co-members, Danica Bonane, Jonaya Bornalo, Francel Del Socorro, me, Sherilyn Pelayo, and Princess Sanahon. What is hexose monophosphate shunt? The hexose monophosphate shunt, also known as the pentose phosphate pathway, is an alternative pathway of glucose oxidation for the production of NADPH and pentose sugars. So, this hexose monophosphate shunt is a metabolic pathway parallel to glycolysis, and in the TCA cycle, or also known as the Krebs cycle, in the oxidation of glucose. It generates NADPH and pentoses, as well as ribose 5-phosphate, a precursor for the synthesis of nucleotides. It has also many other names, like the pentose phosphate pathway, Dickens-Horiker pathway, phosphogluconate pathway, or simply the shunt pathway. It is more anabolic in nature. Anabolic is a metabolic process in which it transforms simple substances into complex molecules, meaning it requires energy to grow and build. And it is not involved in the generation of energy, because in this pathway, no ATP is consumed or produced directly, unlike the glycolysis that produces energy in the form of ATP. And it is about 10% of glucose per day is entered in this pathway, while in the liver and RBCs metabolize about 30% of glucose by this pathway. The purpose of this pathway is to reduce NADPH, the production of ribose 5-phosphate or the RP5, and the production of erythrose 4-phosphate or the E4P. So first is to reduce NADPH. The generation of reducing equivalents in the form of NADPH is used in the reductive biosynthesis reactions within cells. So example of this is the fatty acid synthesis. Second is the production of ribose 5-phosphate. This is used in the synthesis of nucleotides and nucleic acids. And the third is the production of erythrose 4-phosphate. This is used in the synthesis of aromatic amino acids. Where is this hexose monophosphate shunt located? The enzymes of HMP shunt are located in the cytosol. It takes place in the cytosol of the cell, the same location as glycolysis. Cytosol is the fluid portion of the cytoplasm. It is the site of intermediary metabolism, the sequence of chemical reactions by which the cell generates and forms the precursors necessary for biosynthesis of the larger molecules used as the building blocks for cell growth and function. The following tissues such as liver, adipose tissue, adrenal gland, RBC, testes, and lactating mammary gland are highly active in HMP pathway. Liver filters all of the blood in the body and breaks down poisonous substances. Adipose tissue is the energy storage. It also provides insulation and protection for organs, and it is primarily made of adipocytes. And the adrenal gland produces hormones that help regulate your metabolism, immune system, blood pressure, response to stress, and other essential functions, while the RBC carries oxygen from our lungs to the rest of our bodies, and the testes are responsible for making testosterone, the primary male sex hormone, and for producing sperm, while the lactating mammary gland, from the word lactating, it produces and secretes milk. So most of these tissues are involved in biosynthesis of fatty acids and steroids, which are dependent on the supply of NADPH. The hexose monophosphate shunt has two phases, oxidative and non-oxidative phases. So in this oxidative phase, it is reversible. The oxidative word of this phase comes from the process of oxidation. Oxidation means the breakdown of a molecule as it loses at least one of its electrons. So during this phase, glucose 6-phosphate is oxidized with generation of 2 moles of NADPH and 1 mole of pentose phosphate with liberation of 1 mole of carbon dioxide. In this phase, 2 molecules of NADP plus are reduced to NADPH, utilizing the energy from the conversion of glucose 6-phosphate into ribulose 5-phosphate. So in short, it generates NADPH. While in the non-oxidative phase, pentose phosphate is converted to immediates of glycolysis. So, non-oxidative phase is irreversible. It generates precursors for biosynthesis. This non-oxidative phase will be explained further by the next reporter. Step 1. Glucose 6-phosphate dehydrogenase or G6PD is an NADP-dependent enzyme that converts glucose 6-phosphate to 6-phosphogluconolactone. The initial metabolite of the pentose phosphate pathway is glucose 6-phosphate, 2-NADP plus, and H2O. The oxidative phase starts with dehydrogenation at the C1 atom of glucose 6-phosphate, a reaction catalyzed by glucose 6-phosphate dehydrogenase or G6PD. The reaction product is 6-phosphogluconolactone. Conversely, NADP plus is reduced to NADPH during this process. So in this process, NADPH is formed in this reaction and it is a rate-limiting step. Step 2. 6-phosphogluconolactone is hydrolyzed to 6-phosphogluconate by the enzyme gluconolactone hydrolase. NADPH is similar in structure and function as the high-energy electron shuttle NADH mentioned in the cellular respiration. NADPH has an added phosphate group and is used in the cell to donate its electrons just like NADH. Once NADPH has donated its electrons, it is said to be oxidized and is now symbolized as NADP+. NADPH is often used in reactions that build molecules and occurs in a high concentration in the cell, so that it is readily available for these types of reactions. Step 3. Dehydrogenate 6-phosphogluconate to 3-keto-6-phosphogluconate, which is spontaneously undergoes decarboxylation to give ribulose 5-phosphate. The oxidative decarboxylation of 6-phosphogluconate by gluconate 6-phosphate dehydrogenase yields 3-keto-6-phosphogluconate, which is converted to ribulose 5-phosphate, a substrate for non-oxidative reactions and NADPH. So the two most important products from this process are the ribose 5-phosphate sugar because this is used to make DNA and RNA and the NADPH molecules, which help with building other molecules. So, to sum it up, oxidative phase starts with glucose 6-phosphate and ends in ribulose 5-phosphate. First is by dehydrogenation. the hydroxyl ion carbon 1 of glucose 6-phosphate turns into a carbonyl, generating a lactone, and in the process, NADPH is generated. Second is by hydrolysis of 6-phosphogluconolactone by special enzyme called lactone. And the third is by oxidative decarboxylation. NADP plus is the electron acceptor, generating another molecule of NADPH, a carbon dioxide, and ribulose 5-phosphate. To explain the non-oxidative phase, here is Jonaya Bornalo.
Good afternoon, everyone. I am Janaya Di Bornalo, and I'm going to discuss about the non-oxidative phase. The non-oxidative phase is also called the reversible second phase. During non-oxidative phase, pentose phosphate is converted to intermediates of glycolysis. The non-oxidative phase of the pentose phosphate pathway is broken down into 4 steps: the first process, second process, third process, and the fourth process. In the first process, we are going to take in the ribose 5 phosphate or the pentose sugar that is produced in the oxidative phase, which is then converted into ribulose 5 phosphate, which is an isomerase version of it, using the phosphopentose isomerase. The ribulose 5-phosphate undergoes the second reaction with the use of the phosphopentose epimerase, and its product is the xylulose 5-phosphate. The only difference of the two molecules, the ribulose 5-phosphate and the xylulose 5-phosphate, is their stereochemistry. The stereochemistry of their third carbon is different. And why do we want to use the stereochemistry of the xylulose 5-phosphate to the second process? It's because the enzyme of the second process will only going to use the stereochemistry of the xylulose 5-phosphate. The xylulose 5-phosphate will be used in the second process and in the fourth process. In the second process, we will take in again the ribose 5-phosphate and the xylulose 5-phosphate. This process will use this transcatolase enzyme, which requires a cofactor called the thiamine pyrophosphate. What the transcatolase enzyme does is that it takes in 2 carbon groups from the xylulose 5-phosphate and then transfer it to the ribose 5-phosphate, so that the ribose 5-phosphate will be extended into 2 carbon groups, and that is now what we call the pseudoheptulose 7-phosphate. After that, we will take 2 carbon groups again from the xylulose 5-phosphate, changing it to glyceraldehyde 3-phosphate. In the third process, we will use the products from the second process, which is the pseudoheptulose 7-phosphate and the glyceraldehyde 3-phosphate as reactants in the third process. But in the third process, we are going to use a different enzyme, which is called a transaldolase. What the transaldolase does is that it takes 3 carbon sections from pseudoheptulose 7-phosphate, changing it to erythrose 4-phosphate. The 3 carbon sections that we get from the pseudoheptulose 7-phosphate is transferred in the glyceraldehyde 3-phosphate, which is now formed into fructose 6-phosphate. And this fructose 6-phosphate is one of the final products of the non-oxidative phase. In the fourth process, we will use the same enzyme in the second process, which is the transcatolase. we will use again the erythrophosphate phosphate, which is a product in the third phosphate, and again what I've said earlier, we will use the cellulose 5 phosphate that comes from the first process. It is almost similar to the second process. We will take in two carbon groups from cellulose 5 phosphate s- to, to create fructose 6 phosphate, and then we were going to take two carbon groups again from cellulose 5 phosphate to form glyceraldehyde 3 phosphate. This is the net reaction of the process. So as we sum up all the four processes, this is what we actually get. 3-ribose 5-phosphate. The 2-ribose 5-phosphate comes from the first process and the other one comes from the second process. And what we actually get is 2-fructose 6-phosphate plus glyceraldehyde 3-phosphate. These two are the glycolytic intermediates that can be transformed into glucose 6-phosphate. And that glucose 6 phosphate can undergo the oxidative phase to form even more NADPH molecule. So when the cell actually means the NADPH much more than it actually needs the ribose 5 phosphate, we undergo the non-oxidative phase of the pentose phosphate pathway to help us generate even more of these NADPH molecules. On top of that, what the non-oxidative phase of the pentose phosphate pathway allows us to do is it allows us to actually ingest ribose sugars into our body, because via this particular pathway we can actually break down the ribose sugar into glycolytic instruments, and then those intermediates can be used via glycolysis to actually generate high energy ATP molecules. So we see that the link between glycolysis and the pentose phosphate pathway is the non-oxidative base of the pentose phosphate pathway. Significance of HMP shunt, or the hexose monosphate pathway. HMP shunt is unique in generation because it generates important products like pentose and NADPH, which are actually needed for the biosynthetic reactions and various other functions. HMP shunt is also used in the synthesis of sphingosine and cerebrosides. Sphingosine is actually essential for cell growth, survival, migration, immune cell trafficking, angiogenesis, inflammation, and cancer. The next one is the importance of pentoses. Pentoses are used for nucleic acids, ribose for RNA, and deoxyribose for DNA. It is also used in the coenzyme synthesis. Coenzyme molecules are actually mostly derived from vitamins. They are also commonly made from nucleotides such as adenosine triphosphate and coenzyme A. Pentoses is also used in the synthesis of certain vitamins like B2 and B12. Importance of NADPH. NADPH is required for the biosynthesis of fatty acids and steroids. They also used in the synthesis of certain amino acids involving glutamate dehydrogenase, detoxification of drugs and foreign compound, and it requires NADPH. NADPH is also essential to maintain the integrity of RBC membrane and to keep iron ferrous state. Glucose 6 phosphate dehydrogenase deficiency or G6PD. G6PD deficiency is an inherited sex-linked trait. As it is linked to the X chromosome, most people who suffer from it are male. Sufferers cannot make the enzyme glucose 6 phosphate dehydrogenase. This will mean the circulation of sugar in their body is different. Deficiency is seen in all cells of the body but more severe in RBC. G6PD deficiency offers natural protection against malaria as the pathogenic parasites require reduced glutathione for their growth. Individuals with G6PD deficiency are at risk of hemolytic anemia. Hemolytic anemia is a disorder in which red blood cells are destroyed faster than they can be made. Symptoms of G6PDH deficiency includes black urine. And the second one is jaundice. It is a condition in which the skin and the white of the eyes turns into yellow. Third one is reduced glutathione.